No, that first thing I want to tell you that in the morning I was talking about that what is the importance of the renewable energy source, because for that you see that here the global energy potential that has been quoted here in the right hand side, you see the solar energy that is with us about 23,000 terawatt, whereas the world consumption nowadays is 16 terawatts. So it is a tiny fraction of the energy that is really, if we can solar energy, we can nurture in a way so that that we can solve our energy that demand that we require for our day-to-day life. Now the present scenario is that we
Excuse me,
have
sir.
yes.
Sorry, sorry to disturb, but I just want to convey the slides are not moving, I guess. So
It is not moving.
yeah, we are just seeing the introduction slide.
So why it is not
Must
moving? I have to go on to the second slide. I have gone to the second slide. Is not coming. Is the third slide.
no, sir, it's not changing.
So let me. Should I copy again?
No, what you do, sir, you press, you first, what
You
you
see
just
now
close.
you can see.
No, it's not moving.
Oh, I see. So what should we do?
Yeah,
Because
it moved. Yeah.
okay.
Yes, sir. It is fine.
Fine now.
I hope it is okay now. No.
Yes, sir.
Okay, that's fine. So let me let us have should I go to complete the full screen? You see the full screen of whether it is coming or not? Is coming?
Not yet. Not yet.
Oh, then I shall have to go to that other.
Sir,
Okay.
you can do one thing actually. While sharing the screen, you have shared one application window. If you share the entire screen, then in full screen mode also the slide will move, sir.
No, I have
While sharing,
shared. Okay, let me try again
you have
because
shared a particular application only, sir. But you can share. There would be option of sharing entire screen. So if you do that, it would move in full screen mode also.
how to do that actually because full screen. Okay. Is it okay? Hello.
We are seeing slide number 2, sir.
Yes. Is it okay?
Sir, please go in full screen mode. It is still in.
This is full screen now.
Okay.
Now, can you see it?
No, sir, it is not coming. It is coming in the presentation mode only.
But you know that I have given in the full screen.
Please make the slideshow. That might work. Slideshow in the menu bar.
Yes, sir.
Not coming, no. This is slide 3. So let me use this screen, that hub screen. Okay. This is okay.
Yes, sir. Fine.
I think there is some problem. I don't know why you have. I use the Microsoft Word for my regular class, and I don't have any problem. But I don't know what is the reason for that. Okay. Now, what I was telling that the global energy potential that we have, that that solar energy that is with us is about 23,000 this terawatt, and whether our world consumption is about say 16 terawatt. So you see that means the solar energy, if we a fraction, very small fraction, even not 1%, less than 1% we use for, then now we can solve our energy crisis problem. Besides solar energy, we have other renewable energy sources like tide, wave, geothermal, hydro, biomass, and wind that has been given here. And other non-renewable energy sources are natural gas, oil, uranium, and the coal. Now, why I am telling all these things? The reason is that that this is the present energy consumption scenario that we have, that I have taken from IE 2019. You see the 32% of energy that we are consuming from the oil, and 28% from the coal, and 22% from the natural gas. That means most of the energy sources that we are using, that is from the fossil energy, fossil fuels, and 10% is coming from the biofuels or from the waste. That is the present scenario we have. Now here it is similar type of things. Only this pictorial representation is there, because I cannot show you the animation because I cannot have the this slide mode presentation. So another point that I want to stress here, that why we are looking for hydrogen as a renewable energy source. That first is that it contains the high energy density, that means 142 kilojoules per gram, which is quite high as compared to other the fuels that we use in the day-to-day life. And not only that, whenever we use any kind of gaseous fuel or liquid fuel, it is easy for transportation as compared to solid waste, solid fuels. And another positive thing is that hydrogen, if you burn, it produces water, so it is not producing any greenhouse gases, and can be used from the can be produced from the waste, even from the water. So that is why this hydrogen plays very important role. And this hydrogen application is enormous. We can use the fuel cell. It can be used for the generation of electricity, and this hydrogen can be used for industrial use. We have the hydrogen used as a chemical feedstock by the different industry, used for the domestic use for electric power, for the LED light, lighting the LED lights. And the rocket launcher, I told you in the morning that it is largely used in the rocket launcher and different other vehicles, both car, the bus, and the train, water taxi, everything you can use that. Now this is the only one private transport that I have. When we visited Sweden, we observed that they're running with the biogas, and biogas, we know this is nothing but methane. Now here the Tata Motors, they use they recently they manufactured this Tata hydrogen fuel cell bus. So they are using the fuel cell, and the basic fuel that we use, that is the hydrogen, and that is used for running these buses. Now Mahindra and Mahindra, they have come up with 3-wheelers, 2-wheelers, and 4-wheelers, and also there is the other company like Mercedes-Benz, Toyota, all these big company. They are running their 4-wheelers with the help of hydrogen fuel cell system. Now, current technology for hydrogen, that how question comes that how the hydrogen, what is the source of hydrogen? Now, there are different way we can produce the hydrogen. One is called thermochemical gasification coupled with water shift reaction. Then first pyrolysis, followed by reforming of carbohydrate fractions of bio oil. That this is largely used by ISC Bangalore. That Dasapa is using this technology for the production of hydrogen. and direct solar gasification, and miscellaneous that noble gasification process, biomass derived syngas conversion, supercritical conversion of biomass, another is microbial conversion of biomass. So today, since we are more concerned about the biohydrogen production process, so our topic will be related with the microbial conversion of biomass. Now here, I want to point out one thing that biohydrogen, sometimes people also refer that there might be using the thermochemical process for converting biomass to hydrogen. They also consider that a biohydrogen, because since they are using the biomass, truly this is the thermochemical process, that is not a biological process, but they are using the biomass since it is the biological mass. That is why they call it biofuels. So in a true sense, that biofuel is the fuel that is produced from the biomass through the microbial conversion process. Now, if you look at the pictorial presentation, how the different process looks for the hydrogen production, we can divide it into two types. One is called biological, another is non-biological. Now, for first process that we have non-biological process, we use the methane as a raw materials which can be converted to hydrogen and carbon dioxide. And another is the coal gasification process. The coal can be converted to hydrogen. This is largely used. And another is the electrolysis of water that also can produce hydrogen. Another is the photocatalytic reaction through which it can be produce hydrogen. So these are the different non-biological methods that can be used for the hydrogen generation. But we are more concerned about this biological process, and I told you in the morning that biological hydrogen production can be divided into two types. One is called light dependent, another is light independent. Now, light dependent, we have biophotolysis. Again, biophotolysis, you have two types. One get direct biophotolysis and indirect biophotolysis. And direct biophotolysis is usually carried out by the green algae, and indirect biophotolysis is carried out by the cyanobacteria, that is the blue-green algae. I think Professor Ranjana Pandey might have discussed this in details, because she has been working with this process for quite some time. Another is the photofermentation process, where sulfur and non-sulfur bacteria that is used for converting the organic residue like acetic acid, butyric acid, propionate acid to hydrogen. So these are the processes that we have. And here I want to share a short story with you that I had a collaborative project with the Norwegian foreign ministry, and the main purpose of this particular project was to find out the potentiality of this biophotolysis process for the hydrogen generation. And ultimately we have come to the conclusion that this process is no good for hydrogen production, because the rate of hydrogen production is very small, and that is why this process is not very much encouraging for the biohydrogen production. Now, as far photofermentation process is concerned, here at the beginning we don't have any problem, but as the process continues, it produces some kind of red pigments, and also cell mass concentration increases. That causes some kind of shedding effect, and that shedding effect reduces the hydrogen production drastically. So these two processes in the light dependent process is not encouraging as far hydrogen production is concerned. But if you look at the light independent process, one is dark fermentation, another is microbial electrolysis cell. Now, microbial electrolysis cells is a unique device where the waste material can be directly converted to the electricity. Also, it can be used for the production of hydrogen. Now, since we have some kind of scale-up problem, so microbial electrolysis cells is not considered for the hydrogen production. But the dark fermentation process is very easy to operate. It can be operated in two different modes by using both mesophilic and thermophilic. Mesophilic means it is usually that grow at 35 to 37 degrees centigrade, and thermophiles usually grow at 60, more than 60 degrees centigrade. Now, the advantage of using the thermophiles that at 60 degrees centigrade or more than 60 degrees centigrade, most of the pathogens, they are killed. So whenever you use any kind of waste material for this hydrogen production process, the thermophiles, that we can easily allow your desired organism to grow, and it can produce hydrogen, and the kinetics of the hydrogen production also very good. Now question come that when you use the temperature 60 to 70 degrees centigrade, naturally the energy input to the system is very high. Now if you look at the distillery industry, the effluent is coming out at this temperature of 70 degrees centigrade. Now, if we can take this distillery effluent as a raw materials and we put it in the reactor, and reactor, if we put some kind of insulation across the reactor, then we can easily maintain the temperature 60 degrees centigrade, and thermophiles can work there and produce hydrogen significantly. Mesophiles, on the other way, it is very easy to operate, and they are mostly the facultative in nature. The facultative organism has its own advantages, because even the little bit of oxygen present in the system will not affect the system to a great extent. Now, if you consider the obligatory anaerobe, little oxygen may hamper the hydrogen production process to a great extent. So that is why the mesophile and thermophiles that is used for this hydrogen production process. So this I am going to discuss in details. Now, when you talk about the biological hydrogen production process, three things that is very important. One is called essentiality, the constants in the conventional production techniques that is to be overcome. Because if you look at the thermochemical gasification process, that is usually take place at a high temperature and high pressure. So this our biological process usually taking place at the low temp, as the ambient temperature and atmospheric pressure. That is why that high energy intensive situation can be improved. And versatility that offers the potential production from the variety of these renewable energy sources, particularly through the bioremediation of refuse and waste, because waste is kind of the nuisance to the environment. So if we can, we shall have to treat the waste material, because we cannot allow our environment to deteriorate. So naturally, if we use this waste material and produce hydrogen, one way we are generating the energy, another way we are saving our environment to a great extent. Another is the naturality that near zero pollution and cost-effective process due to simple technique. That is the advantage of this process. Now, these various approaches that we have already discussed. One, this is called direct biophotolysis with green algae that is involved. How it is, this is photosystem 2, photosystem 1, both are involved in the system. And indirect biophotolysis, that is cyanobacteria that is involved, how this take place, that has been shown here. And photofermentation process at the bottom has been shown here. And dark fermentation process is pyridoxine that take place, and hydrogenous enzyme and formate lyase, those enzymes taking the major role for the hydrogen production. Now, this is the different use of this. This is the different flue gas can be used for the generation of this green algae, and this green algae can be used as a nutrient supplement for the. I told you in the morning that when you use any kind of waste for biological system, that we shall have to supplement with some kind of nutrients, because you know, when the microorganism grow, it required carbon source, it required nitrogen source, it required minerals, and it required vitamins. So naturally, when the wastewater or waste we use, that has lack of this nutrient. So this micronutrient you might have to use. If you use in the form of some kind of waste material, that will be better. So algae can be used, and water hyacinth algae, those can be used as a nutrient supplement for the better utilization of the waste material. And raw wastewater different by the industry, that can be used as a substrate for the hydrogen generation. And then ultimately this hydrogen you can use for the that microbial fuel cell, operation of the microbial fuel cell. Now this is the metabolic pathway, how hydrogen production take place. You can see that carbohydrate that reach substrate, maybe suppose if we talk about the cellulosic material, if you talk about the starchy material, that first undergo the hydrolysis process, and through hydrolysis process we produce this glucose, and this glucose, when it passes through the Embden-Mayer pathway, it produces the pyruvic acid, and this pyruvic acid, when it follows the acetate pathway, it produces the 4 moles of hydrogen. But if it follows the butyrate pathway, it produces the 2 moles of hydrogen. But if it produces follow the lactic acid formation or ethanol formation, it doesn't produce any kind of hydrogen. In other way, it consumes a lot of NADH, which is very much essential for the hydrogen generation. So then formate also plays very important role for the hydrogen production. So this is how the metabolic pathway looks, and that how the hydrogen production takes place. Now I was talking.
Excuse me, sir.
Yes.
Sir, can I ask you a question in between or at the end?
At the end, you ask. I think it will be better. Otherwise, I may. Because time given is 1 hour 30 minutes.
Sure, sir.
So if it goes to 2 hours, then it might be a problem for you.
Sure, sir.
So at the end, I can take all the question one after another, and I explain more details. Okay.
Sure.
And now I cannot explain more details because I still have to cover all the things. Okay.
Okay, sir.
Okay. Okay. Thank you. Now, advantage, I was talking in the morning that biohydrogen production process as such may not be attracting, because when you compute that, you see in the bottom that if we consider the acetate pathway and we observe that 1 mole of glucose can produce 4 moles of hydrogen, and if we consider the 4 moles of hydrogen, our energy recovery is 34.2%. That is the maximum amount of energy we can recover. But if you look at the same time, if you look at the biomethanation process, we see that 1 mole of glucose can produce 3 moles of methane, which can produce, with the energy recovery is 85%. So if you look at if you compare with this process, naturally that your biomethanation process is more attractive as compared to that of biohydrogen production process. Now, there is a very interesting thing here I want to share with you, that if you look at the anaerobic digestion process, that is usually performed by the 2 group of microflora. One is called the acidogens, another we call methanogens. Now acidogens, that usually what they do, they convert the organic compound to the volatile fatty acid. And what are the volatile fatty acid? The acetic acid, butyric acid, propionic acid, valeric acid, those are the volatile fatty acid. They form, and this formation of this organic compound like cellulose, starch, glucose, sugar into this volatile fatty acid usually performed by the acidogens, and methanogens, they convert this organic acid to to methane and carbon dioxide. Now, now since the two group of microflora present in the biomethanation process, so always there will be inside the reactor, there will be tussle between these two groups of microflora because they are differ from each other with respect to their substrate requirement, with respect to their nutritional property. All these things, they are differ from each other. So it is always better if we can separate the acidogens and methanogens so that they can work in a better way. And because in a reactor, if your rate of acid formation is more as compared to methane generation, the pH will go down, and naturally you will find that your methane production will be stopped. This is the scenario that we have. To avoid that kind of situation, always you have to maintain the some kind of balance between the acid formation and the methane generation. But as soon as you separate the acidogens and methanogens from each other, then you can easily control the system. You can in one reactor you can produce as much as acid you can do, and you can add the acid in the methanogenic reactor in a manner so that it can be converted to methane and carbon dioxide. So now the way I am sharing with you all these things, these acidogens which produces the organic acid, they are responsible, we have observed, to produce hydrogen. And we explored that what is the potentiality of these acidogens for hydrogen production. Since we have been working with this hydrogen generation process for more than a decade, and we found out these acidogens are very good for hydrogen production. So this anaerobic digestion process, what is in practice, what we can if we have in two reactors, one reactor we can use the acidogens, and then we can generate the hydrogen, and the reactor, that organic acid you can convert to methane and carbon dioxide. Now in the bottom you see that if you segregate the process in the two state, then our energy generation is 91%. So naturally the overall energy generation increases when you go for the biohydrogen production process. Now this is how schematically the process can be represented. That stepwise, that complex polymer, we have polysaccharide, we have proteins, we have lipid, and this usually first is this pre-treatment, undergo pre-treatment or hydrolysis, where it produces sugar, amino acid, and long-chain fatty acid, and at the same time it produces hydrogen and carbon dioxide. And this organic acid, when it produced during the organic acid production process, and then this organic acid is converted by methanogens, by methane and carbon dioxide. So this is a better avenue for the hydrogen generation. Now, as far our research work is concerned, how we have, this is a kind of journey that we have for this biohydrogen production process that we have developed at IIT Kharagpur. That I want to show you that how we have done that. When we started this research work in the year 1999, and we tried to find out that what are the processes are available for the biohydrogen production. And when you review the biohydrogen production processes in 1999, we observed much emphasis has been given on the photobiological system, because scientists, I told you in the morning, scientists initially they thought that that algae has the capability to hydrolyze the water into hydrogen and oxygen. This might be the best route for the hydrogen generation. But when they started using this, then they observed that this molecular oxygen, whatever is produced, that gives some kind of inhibitory effect on the iron hydrogenase enzyme that is involved for the hydrogen generation. So that your hydrogen production is greatly affected. So what we have done, we try to find out, other than photobiological process to hydrogen production, So we try to isolate the bacteria from the environment, and in process we identify interbacter clocking from the leaf of some flower plant, local flower plant. Then this name of the organism is Interbacter clocki IITBT08. We find this organism has the capability for producing hydrogen very high rate and very fast. Sorry. And this organism, then we try to establish the fact why this organism producing this hydrogen so fast. And we find the format lyase is the major enzyme present inside the organism that plays important role for the hydrogen generation. So what we have done some molecular cloning, characterization and overexpression of the noble iron hydrogenase, whatever is present there, redirection of metabolic pathway, because I have already shown that how the hydrogen production takes place, and we have observed that if the carbohydrate it goes for lactic acid formation or methane or ethanol formation, there is no hydrogen formation. But in other way, they consume some kind of NADH for during this lactic acid and the ethanol production process. So what we try to do, if we can close this pathway, then through the mutant development of some mutants, then it is possible to have more NADH in the systems, which can use as a currency for the hydrogen generation. Then we have studied the optimization of the physicochemical parameters for the hydrogen production. And when we study all these things, once we observe that this study is mostly carried out in the batch system. Now, batch system is no good for the hydrogen production. The reason is that if you go for the commercialization process, we observe the batch system is the most inefficient system, because maybe due to the reason is that the rate of reaction is very poor, less, and not only that, you have some downtime which is required during the batch operation processes. So naturally you have to go for the continuous production process, and we develop some kind of wholesale immobilization system for the continuous hydrogen production. This has been done both in the 20 liters and 800 liter reactors, and we successfully operate this system for quite long time. Simultaneously, we work with modeling and simulation of hydrogen production and effect of iron on the fermentative hydrogen production we studied. And finally, we studied the hybrid bioreactor system. This hybrid bioreactor means, that at the end I have discussed that there are different way we can operate the hybrid system, where hydrogen production followed by methanation, hydrogen production followed by microbial fuel cell, hydrogen production followed by the bioreactor, fermentation process. So different type of hybrid system. We try to work with this and try to find out which process will be suitable for the hydrogen generation. And ultimately, we operated and one pilot plant, or 10,000 liter pilot plant at IIT Kharagpur, and also my another collaborator, Dr. S. Venkatamohan. I think he will be talking in this particular lecture series that he has established that 10,000 liter reactor at IIT Hyderabad. So but that the process, the two processes are little bit different. Our process mainly target the industrial wastewater, and his process mostly target from the food waste material that they have considered as the raw materials for the hydrogen production. This is how hybrid system looks like. That I was talking about dark fermentation and photo fermentation will be when you combine, we call it hybrid reaction. We have direct biophotolysis and direct biophotolysis like this. Now, The dark fermentation process, when you talk about the biomass, agricultural product, and the organic waste generated by the different industry, or the municipal solid waste also is the organic waste, and this is undergo pre-treatment, and then we fermented by the metabolic engineering organisms, and we produce hydrogen and carbon dioxide. This is how it can be explained. Now, as per substrate is concerned, that different type of raw materials can be considered as a substrate for this dark fermentation process. One is carbohydrate. Carbohydrate may be of different types. One is glucose, sucrose, maltose, cellulose, and cane molasses. Cane molasses contains about 50% of sugar. And cellulosic biomass, we have energy crops like corn, wheat, straw, and paper pulp. And waste, we have municipal solid waste, food waste, industrial waste, and the wastewater. So different type of this raw material can be used as a substrate for the dark fermentation process. Now, if you look at the waste availability of India, it like this. We can broadly classified into two types. One is called solid waste, another we call liquid waste. Now, solid waste can be divided again into three types. One is called municipal waste, and there is the agricultural waste, and there is the industrial solid waste. The municipal waste, we have organic litter, and the household waste, and the sewage. And agriculture waste, we have algal biomass, energy crops, food crops, and lignocellulosic biomass. Industrial solid waste, we have food processing industrial waste, dairy waste, dairy industrial waste, and the agro industry waste. Now, liquid waste, we have two types. One is called industrial wastewater, and there is domestic wastewater. Industrial wastewater, we have distillery effluent, we have paper mill effluent, we have starchy wastewater, we have pharmaceutical wastewater. The domestic wastewater, we have canteen waste, we have household wastewater, and the starchy wastewater. Biomass, the boon to the society. That means that what I want to say, that biomass is considered to be one of the key renewable resources of the future, both at the large and small scale operation, because this is very important. Because when you talk about any kind of chemical process, that cannot be operated in a small scale. Always you have to think for the big scale. But in the biological process, always you can operate both in the small scale and big scale. Because I can give a very typical example. The 10,000 liter reactor that we have established at IIT Kharagpur, and we found the amount of hydrogen we produce per day that can give light to a small village. Because if we pass through the fuel cell and generate the electricity, that can illuminate this one small village. So this is the small scale application. Now, when you go to the industry, because this technology has been given to one industry, that is the Dhampur sugar mills, and they are now working in the commercial scale, and they are going to club with the biomethanation process and to make the process successful. It is already supplied the 14% of world primary energy consumption. This is already fulfilled. That is that 14% energy that consumption can be fulfilled from that. Now, if you look at this in the developing country, this biomass utilization is more. Even in some cases, it might be 90% in some developing country. Poor country, they might be using this biomass as a source of energy. Now, when we talk about the microorganism, up till now I was talking about the raw materials. Now, when you talk about the microorganism, which take part in this particular fermentation process, I told you it broadly divided into two types. One is called mesophile, and another is thermophiles. Now, in mesophile, we have two types. One is called obligate anaerobe, and this is facultative anaerobe. Obligate anaerobe, we have Clostridium species, and facultative anaerobe, we have Enterobacter species. Here I want to stress the facultative anaerobe is very easy to handle, because even little trace amount of oxygen present in the fermentation broth, it will not affect the process to a great extent. And thermophiles also it carried out by the two groups of microflora. One is called obligate anaerobe, another is the facultative anaerobe. Now, obligate anaerobes we have Thermotoga, and the facultative anaerobe is the Klebsiella. Now, this is the history of the biohydrogen production process. If you look at the Heinz Graffen, the scientists of University of Chicago, observed that green algae has the capability for producing hydrogen instead of oxygen. But that is the starting point of the biohydrogen production process. Later on, in 1990, the work of from California University observed that production of hydrogen from algae in sulfur deprived media, because here we can use the organic material as a substrate for the hydrogen production. The graphene used the water, but here is the organic material. And later on, my friend John Brenneman, he published one paper that is in Nature Biotechnology, and where he claimed that biohydrogen required serious funding for its improvement. Now, this is the roadmap that we have that on the biohydrogen production process we have. Now, I am glad to report you my H index is 55 now, and my I10 index is 144. Now, as I told you, we started the research work in 1999. The first paper we published in 2000, Enhancement of Hydrogen Production by Interbacter Clocky IATBT08. This is published in Process Biochemistry Journal. And then we published and the one review paper in 2001, that hydrogen production by biological processes, survey of literature, and with Professor Veziroglu, who is considered as the father of hydrogen in the world, because he brings this hydrogen technology to the world for the mass scale applications. So I have a very great opportunity to work with them, and I'm glad to report you the citation of our paper is more than 2,000, and so this is the highest citation that we have as per biohydrogen production processes concerned in the literature. And then we also work with the hydrogen production process that, as I reported, that we published in Energy and Microbial Technology. Then I told you we did some kind of molecular biology work that we published in BBRC. Then we also observed that how the hydrogen partial pressure affects the hydrogen production process is published in Biotechnology Letters. And later on, we try to find out the different types of bacteria from different sources, how it affects the hydrogen production. One example is the Bacillus coagulans, which we isolated from the anaerobic Suez sludge, and this we published in the Bioresource Technology Journal. Then some advances process also we work with that, and then we use some photobiological process that we published in International Journal of Hydrogen Energy. The effect of pH, how regulated pH influences this fermentation process that we discussed. The thermophiles, how affect with thermophilic mixed culture we use just to find out that how it is good for the hydrogen generation. This we published in International Journal of Hydrogen Energy. Then the continuous thermophilic biohydrogen production, this we published in Applied Energy. And then we use the mixed consortia, different type of consortia, mixed consortia, synthetic co-culture and the pure culture, how it can be used for the degradation of distillery effluent for the hydrogen generation. This we published in Bioresource Technology Journal. Genomic proteomics that has been published by Pallavi Sinha and others. The Pallavi Sinha is one of the speakers in this lecture series, so she can share with you that her experience on this hydrogen generation process. Then one interesting things we we try to work with that that there is the called acetone butanol fermentation process. In the acetone butanol fermentation process, the first step is the acid generation, and second stage is the that butanol formation. So here, this process also we try to explore how it can be used for the hydrogen generation and biobutanol generation simultaneously for maximizing the energy product formation. Then recently we published another paper that is on this commercialization of biohydrogen production process from distillery effluent, and this the lecture that I have given today, that is the holistic approach from lab scale and to pilot scale. This has been published by International Journal of Hydrogen and Energy in 2020. And recently we published another paper on optimization of simultaneously butanol and biohydrogen production. This also we published by International Journal of Hydrogen Energy. So this is a long journey that we have on this biohydrogen production process. Now, if you look at the microorganism that we use in this particular fermentation process, the morphology of this organism is like this. Enterobacter is kind of rod-shaped. You can see it's the rod-shaped of the organism. Klebsiella also more or less rod-shaped. The Citrobacter friendly we isolated from high oil containing soil, and the Clostridium species we isolated from the anaerobic digester. Clostridium acetobutyricum also from anaerobic digester. Bacillus coagulant we isolated from the Suez sludge, and acidogenic mixed consortia we isolated from the anaerobic digester, and thermophilic, the thermoacetobutyricum that we isolated from the anaerobic digester. So these are the different organisms we try during our research work, and this is the kind of the comparison of our research work as compared to reported research work. As for example, that Enterobacter clocky IATBT08, you see the yield is about, when you use the glucose as a substrate, it is 2.2 moles of hydrogen per mole of glucose, but when sucrose used as a substrate is 3 moles of hydrogen per mole of sucrose. But when you use the double mutant 11, then it is 3.8 per moles of glucose. So this is almost close to 4, what we have observed during the acid pathway, acetic acid formation pathway. This is the details of the Enterobacter clocky that we have been working, and we are quite successful use this organism for this hydrogen generation. And this is kind of experiment, the experiment that we have carried out in the lab. This is the control bioreactor. But we not only use the control bioreactor, we use the several custom-made bioreactor also. The main advantage of this control bioreactor is that you can optimize different operational parameters like temperature, pH, and agitator speed. All these things that you can easily control very easily. And this is the control module. In the left hand side, you see this is the control module, and right hand side we have the gas collector. And this gas collection takes place by the water displacement method. And this is kind of profile that we have, how the cumulative hydrogen production take place. This is the cumulative hydrogen production take place, and we find that maximum our hydrogen production take place within the 11 hours of fermentation process. It's quite fast. If you look at this photobiological process, it takes couple of days, and that too the amount of hydrogen production is quite less. So this is the advantage of this process, that the reaction is very fast, it produces, and this organism has the capability of use the different type of carbon source, cellobios, arabinos, that fructose, maltose, potato starch, CMC cellulose, xylose, dextrose, and sucrose, and all the yields is given here. Now this I want to share with all of you. This is very interesting, that when this Enterobacter clocky use both the condition, one is the aerobic condition and this anaerobic condition. Now, if you use the aerobic conditions, then it up to 10 hours of fermentation, that pH decreases, then all of a sudden it increases. And during this aerobic fermentation process, no hydrogen production will take place. But during anaerobic fermentation process, you see, even after 10 hours of fermentation, pH decreases. And this helps you for the hydrogen generation. This kind of pH profile we observed, and we find 6.5 media initial pH of the media is find most suitable for the fermentation process. Now another thing that we work with the continuous system that I mentioned before, and we use some kind of immobilized solid matrix. Now inside the things you can see the coconut coir we use as a solid matrix, and coconut, why we use the coconut coir, because the surface area per unit mass of the solid matrix is quite high as compared to other lignocellulosic material, like we use the wooden chips, we use the this rice straw. So we find that coconut coir is better as compared to other this we have shown in the letter. And beauty of this that you can fix the cell on the solid matrix, and you can pass your substrate from the bottom and take out the liquid from the top, and hydrogen you can collect it like this. So you see that this is the different type of solid matrix we have used, SMA, SMB, SMC, and performance is leading there. The SMC is nothing but coconut coir, where the cell density, you see cell loading, gram of cell per liter of the reactor is quite high. It is 44 grams of that dry cells per liter of the reactor. This is quite high actually. And then the major problem of this particular immobilized system is the gas holdup problem. And this can be they overcome by using different type of reactors. We use the tubular reactor, we have taper reactor, we have rhomboidal reactor. The rhomboidal reactor has advantages as compared to tubular and the taper reactor. The reason is that if they have the convergence and divergent section. You can see at the bottom they have convergent and divergent section. So when the gas production takes place during the convergent section, the bubbles they cluster together, and then when they make a big bubble, and then they give the buoyancy force and will go up. And due to this higher force, there will be some kind of excitation that takes place inside the reactor that has better conversion efficiency. So that is why at the top you see, as compared to tubular reactor, rhomboidal reactor, your hydrogen production increased by 13.6%. And then this when we operate in a continuous system, we try to find out what dilution rate it gives the maximum amount of hydrogen production. We find 0.45 hour inverse. 0.45 hour inverse means hydraulic retention time was the close to 2 hours. It's a little bit more than 2 hours. That is good enough for the hydrogen production. So you, as compared to batch process, I told you in the batch process we required 11 hours. Now this, when you operate in the continuous system, you can reduce to 2 hours or little bit more than that. So your rate of reaction will be quite fast. So now this is kind of this hydrogen production by the immobilized system that we have reported how it is continuous production with respect to different dilution rate, how its production takes place, how pH profiles changes. All this thing has been reported here. And this is a very interesting curve, because that generation time of the free cell is usually low as compared to immobilized cells, because when cells are supported or is immobilized on the solid matrix, it is expected their growth rate will be little bit hampered. But we have observed when you use the coconut coir, we observed that that generation time almost close to that of the free cell, even a little bit less than that. Maybe due to the reason that the coconut coir might be having some kind of that good effect on the substrate conversion process or on the microorganism. That is why we get the better, less hydrogen that generation time as compared to this hydrogen. Now, how we calculate, the question comes how we calculate the generation time. Between the two peaks is considered the generation time, because when you this way how we monitor, because we continuously passing the liquid through the reactor, am I right? And when the daughter cell formation is there, take place, we make the flow rate in such a way that whatever excess cell is formed, that is coming out from the reactor. So in the outgoing liquid, when you monitor the cell mass concentration, that will give you time to time what is the how much cell is coming out. So you know that, so we can easily find out that two peak position, we have find out that when the peak to this peak height to this peak height, this difference will give you the generation time of the immobilized cell. And this is how the cells are immobilized on the surface. This is the coconut coir. You can see the coir. You can see the thread-like structure you can have. And this is the white type of things on the surface of the coconut coir, where you can see how the cells are immobilized on the surface and how the cells are oozes out from the surface. That bottom part you can see that how cells are oozes out from the surface. That you can find it out. Very interesting observation that we have. The another important research work we try to carry out, that is the effect of partial pressure on the hydrogen production. Then we have the Lee-Catlier principle. You see at the above there is the equation that how the hydrogen production takes place. That 1 mole of glucose usually converted 2 moles of acetic acid and 4 moles of hydrogen. And your Lee-Catlier principle, that equilibrium constant equal to concentration of product divided by concentration of the substrate. Now, since our product is the gas, so if we use some kind of vacuum, you can take the gas out. Now, if the product concentration reduce, then what will happen? The more substrate, because Kc is constant, the more substrate will convert it to product, so your gas production increases. That is exactly we observed during our research work, that we have find that you see that 760 millimeter mercury pressure, we have this hydrogen yield is 1.9 moles of hydrogen per mole of substrate, whereas 380 millimeter of mercury pressure, it is 3.9 moles of hydrogen per mole of substrate. So it is, and but if you decrease the pressure further to 330, then your hydrogen production decreases. That might have given some kind of bad effect on the microorganism. So we apply the vacuum in between 760 to 380 millimeter mercury pressure just to enhance the hydrogen production. And this is we observed that how the cell density also changes during the vacuum recycling. And this is the summation of the work that we have done, that the Intravector Clocky and Intravector IIT BT08 and DM11, and that 3.8 we got. This is the yield we got, as compared to the other microbial cells that is reported in the literature. This is quite attractive. Now let me come back to the present status of our activity that we have. Now presently we are working with different type of waste material. We considered the lignocellulosic biomass. We considered distillery effluent. We considered starchy wastewater. We considered some kind of algal biomass which grown on the wastewater. We use also some kind of residue that is the cane molasses, and the different type of organisms that we have already reported before. And as I told you that whenever you use any kind of waste material, it has lack of nutrients. So what we have done, we try to use some kind of defatted de-oil cakes, like groundnut de-oil cake, coconut de-oil cake, mustard de-oil cake, sesame de-oil cake, rice bran DOL cake and soybean DOL cake. Different. This is the waste product of the different well making industry. And if you look at the composition of the wastewater, the distillery effluent and starchy wastewater is giving here. And here I want to point out the COD value of distillery effluent is about 58 milligram per liter, gram per liter, and whereas starchy wastewater is 25 gram per liter. But whereas cane molasses is quite high, it is 900 gram per liter. So other composition is given here, and we try at the same time we try to find out that what is the composition of the DOL cake, that is the groundnut DOL cake, coconut DOL cake, mustard DOL cake, then the soybean, sesame, rice bran, and water hyacinth. And we have different analytical data is given here that you can see that. So the beauty of this that it contains a lot of protein, and that inside this protein also required for the growth and for the growth of the cells, and also it has some kind of minerals and vitamins which help for the growth of the model or growth and of the organism. Now this is we try to find out that how it is comparable. This is the intervector clocking. When we use the cane molasses, we use traditionally we use the yeast extract as a nutrient supplement, and this we get that cumulative hydrogen production is 1.9, but when we use the groundnut DOL cake, then we find it is 2.8. And that DDGE, this is the distiller grain solid. This is that is obtained from the IFB agro industry. That is also kind of waste product. This is 2.5. So distillery effluent also we get this kind of results. So if you go through this table, you will find that that as compared to the conventional nutrient supplement, that this extract, this material, that the DDG and or the groundnut DOL cake, that is quite attractive. Not only attractive, this gives the better results. The cost of production process is drastically reduced. Now, this metabolic fermentation process, we operated in 20-liter reactor. If you visit my website, you can find out this video of the operation of 20-liter reactor, and also you can find out the video for 1,000-liter reactor, that you can easily find out the what I can isolate. I can report here that 20-liter reactor we can produce about 68.7 liters of hydrogen per hour. This is quite high because that. And here you see that this is the custom-made reactor that we have done. We are designed by ourselves. We fabricated locally, and that the middle we have this vacuum recycle system that we have developed by ourselves, and the right hand side we have the gas collector. So this produces that this much of that Now this again we try to operate in a controlled fermenter. This is also 20 liters, and try to operate by using the previous system we operated by mesophiles. This we operated by thermophiles. This produces distillery effluent. We produce 2.9 liters per liters, and here it is cane molasses is 4.8 liters per liter. That means if you multiply it by 20, it will come around say about 96 liters of gas will be produced from this system. Is quite high because this is kind of comparison that we have with the different type of substrate, because we use without supplement cane molasses. If we use as such as a raw materials, how much hydrogen production is taking place. If you use the yeast extract, how much hydrogen is produced, and if we use the inorganic source, the diammonium phosphate, how much hydrogen is produced, and in the groundnut DOL cake, how much is produced. Similarly, we have carried out the experiment with the distillery effluent. We carried out the experiment with starchy wastewater. And different type of kinetic I mentioned during my presentation that we did a lot of that modeling and simulation study that bioresource technology journals. We published one paper fully on the modeling and simulation of the biohydrogen production process. Anybody interested to know this modeling and simulation of the biohydrogen, please go through those papers. I think it is you'll get the information. So here we use the different models. We use the logistic model. We use the first order reaction kinetics. We use the part model. We use the modified Gompertz equation. We have used the Ludeking-Pirate model. So different type of constants we have calculated from this system that has been discussed here, that both with the treated and untreated the material that we use as the feedstock, that the GDOC we use as a substrate, and then we considered kind of treated and untreated GDOC. You see that when untreated you have very hard structure, and when you treat with acid we find some hole has been developed, and that increases the susceptibility of the solid material for the enzymatic attack, that increase the conversion process to a great extent, and where the CRI value, the crystallinity index value increases by 25%. And this is the CSTR operation that we have carried out. I told you, we not only use the control bioreactor, we use the custom-made bioreactor. This is the circulated water bath that is used for maintaining the temperature. We have magnetic layer at the bottom to make the homogeneity in the reaction mixture. So this is kind of profile that we have, and what we observe, the maximum 870 milliliter of hydrogen is produced per liter per hour was achieved as a flow rate of as a dilution rate of 0.4 hour inverse that we observed. And then we also carried out this system, the 20 liter system I have already mentioned, and this is the vacuum recycle system that we developed by ourselves, and this is how the cells are immobilized on the surface of the solid matrix that I have shown here. And this is the continuous hydrogen production using 10 moles in the extracted immobilized whole cell bioreactor. And we find that different type of metabolized formation is there, acetic acid, butyric acid, that formation is there. This is how with the dilution rate, how different metabolite formation is there, that also we try to monitor. Now, this is kind of material analysis of the 20 liter reactor. We find that if we start with 1 kg COD, we able to get hydrogen about 81 gram of COD in the form of hydrogen, and remaining we have in the form of metabolites and also biomass and effluent that we have. So it contains the significant amount of volatile fatty acid. You see, acetate is 31.3 gram, butyrate is 75.9 gram. It is quite high. So this can be easily used by the methanogenic reactor to convert it to methane and carbon dioxide. So this is the system that we have. Sorry, this is the going. And this is the recently we worked with a USB reactor. The USB reactor is the upflow anaerobic sludge pump reactor. Hello. Can you mute your mic? Hello. Can you mute your mic, please? Hello. Hello. Hello.
It will call you now. You're there, na?
Please mute your mic.
Stop and they'll invite.
Okay.
They'll call if any
I
staff.
think organizers should mute other mic, because you can do that. You know that, please.
Go and show your video. They are taking snap. போய் வீடியோ ஆன் பண்ணுங்க போங்க.
Hello. Hello, can you hear me? Hello. Anybody there?
Yeah, we all are there, sir.
अरे भाई वहाँ पर क्या बात होता है, because you should control this somebody disturbing, Okay, let me share our recent work that we have done. That is the USB reactor, that upflow anaerobic sludge bucket reactor. At the bottom you can see that the reactor is given there, and this is USB reactor largely used in the biomethanation process for the high rate methane production. This is largely reported, but we want to explore how this process is suitable for the biohydrogen generation. And major problem of using this USB reactor is the development of the sludge bed. That requires a couple of months just to develop the sludge bed. As you know, that anaerobic organisms, they are very slow growing. They are not fast growing. So it will take quite long time to just to develop the bed. So this is in the top you will find the performance, how the sludge bed that increases with respect to time, and then the right hand side we have mentioned how the hydrogen production takes place. That is also it increases with respect to time. And our results is we get very encouraging results. That is 12.3 liters hydrogen per liter per day, which is very high, and with a hydraulic retention time only 3 hours. That is we observe. This is quite high as compared to that other reported process. So now this is the 50 liter reactor. We try to operate. This is the 50 liter reactor, and we get about 2.9 liters of hydrogen per liter was achieved with a yield of 12.25 moles of hydrogen per kg of COD. And then this is the biohydrogen pilot plant that we have established as the IIT Kharagpur, and this is our pilot plant that is in operation. The video demonstration of this process has been available in the in my website www.bio2iitkgp.in. I request that Professor Ranjana Pandey, if time permits, they can show this video. This video is only for 16 minutes, and this gives a very good idea that how we operate the pilot plant. And this is the kind of the pilot plant we have developed. You can see in the left hand side we have 10,000 liter reactor, and in right hand side we have three cubic meter reactor, 3,000 and 3,000 liter reactor. These two reactor at the back, this is considered as the feed tank, and at the front you have this 500 liter reactor. Then actually we started with 5 liters and in the sake plugs, then 5 liter to 50 liters, then 50 liters to 500 liters, then 500 liters to this half of the this reactor we fill up with 4.5 cubic meter of media, and in the industrial effluent, then we put the 500 liter of the culture, then we make it to 5,000 liters, and then when the rate of hydrogen production is maximum, then we fill it up to 10,000 liters, and we produces that about 76 cubic meter of hydrogen per day we produce in this plant. The huge quantity of hydrogen, and we have calculated if this hydrogen we can pass through the fuel cell, it can give the light to the small village. And this is the kind of that development we have. We are small scale. Our cumulative hydrogen production was quite less. It is 2.6 liter per liter. As we scale it up, it is 2.9, and then finally we get 7.6 liters. So scale up always gives the better hydrogen production as compared to the small scale operation. And this is kind of the energy analysis we have done. If we start with 100 kg of cane molasses, we find our energy recovery is 21.6% with respect to substrate added, and substrate remove is 37.9%. So again, I am saying that we try to compare our results with the report that is reported by other groups. That we find that as per other groups, our results is quite interesting. We got the maximum hydrogen production is 18.76 moles of hydrogen per kg of COD remove, whereas this other is quite. But Rain et al. They have reported 26.36 moles of hydrogen per kg of COD remove. But that theoretically maximum hydrogen production we get 20.8 moles of hydrogen per kg of CO2 removed. How we got this 26.13, I could not understand. So this is under question actually. So this is the 3 books we have published that on this biohydrogen, related to the biohydrogen production process. First book we published in the year 2014. This with my student Namita Khanna and my postdoc, that is Chitralekha Nath Dasgupta. Then second book we published recently, the 2020. This is Fundamentals of Biofuel Production Process with Jhansi L. Banarasi. And then we published another book that is with Shantanu Rai, that is Biohydrate, the fuel for the future. So these are the 3 books, and this can be used as a textbook because if anybody interested on the bioenergy generation process, this book gives you the detailed information on this process. Now at the end, I want to discuss about the integrated process, what you call the hybrid process. I told you this is the hydrogen generation process, and it can be clubbed with biomethanation, it can be clubbed with the acetone butanol fermentation, it can be clubbed with photofermentation process, it can be clubbed with bioelectrochemical process, and it can be clubbed with the microalgal cultivation process. So all these processes, we find among all these processes, the biomethanation process is more attractive as compared to other processes. The integrated system development to maximum the substrate energy recovery, we have biohydrate process, hydrogen followed by methane. We have biohydrate process followed by microbial fuel cell. We have biophasic process, that hydrogen followed by biomethanol. The maximum gaseous energy recovery 44.8% was observed by integrated biohydrogen production and the biomethanation process that has been reported by my student Sinu Kumari. And overall energy recovery is 85.6% was obtained by the biohydrogen biomethanol process reported by Vaisali Singh, and the overall energy recovery is obtained about 60% the biohydrogen followed by microbial process reported by the Vaisali. So I am thankful to all my sponsorers, particularly Department of Biotechnology, Government of India, Ministry of New and Renewable Energy Sources, and then Defense Research and Development Organization, Department of Science and Technology, National Science Foundation USA, and German Academic Exchange Service. The major collaborating institutes are there: University of Miami, Oslo University, Uppsala University, Denmark Technical University, Ruhr University, IICT Hyderabad, TERI, JNTU. Also, I have the collaboration with Allahabad University, where Professor Ranjana Pandey was there as a faculty, and then we have banners in the university that also they are my collaborator. My hydrogen group, you can see that we have our girl students are most dominating as compared to the boy student, and though the research work on hydrogen started by Narendra Kumar, and this button was taken by all the researchers, and try to make this process a success. And we are thankful. I am very much thankful to all my PhD students who contribute, PhD and postdoc, because Pallavi and Chitralekha is my postdoc in my lab for quite some time, and their contribution that lead to our biohydrogen production up to this level. Thank you very much. If you have any question, I'll be very happy to answer. Thank you.
Now the platform is open for discussions.
Well, some student has some question at the beginning. I want to take those questions first. Hello, student.
Yes, if you have any query, please ask.
So what I can do, Anjana, that if they have any question, they can send me the question in the email. I try to answer it. Even they don't have any question now, I can give the answer. I don't have any problem. So that, and I request all of them to visit my website and see the video. Then you will get the information in details. Okay.
Okay. Thank you, sir. Thank you so much, sir.
Thank you for inviting me and sharing my thought. Okay, I wish your work, your program should be a great success. Thank you.
Thank you so much, sir. Thank you so much.
Thank you.
Thank you, everyone. This is the end of today's session. Links for tomorrow's session will be provided to you on mail and on the WhatsApp group.
Thank you.
Okay then.
Thank you.